G'day, science enthusiasts. Let's take a genetic journey from nuclein to genome mapping. Now, here's a common misconception that plagued the early days of genetics: the confusion between genes and chromosomes. Humans have 46 chromosomes organized into 23 pairs of alleles. But these chromosomes house a vast number of genes, tiny packages of genetic information. Let's wind back to the 1860s. Frederich Miescher hypothesized the existence of chromosomes and genes, predicting they'd be made of a substance he called nuclein. In 1869, he isolated this material. Fast forward to the late 1950s, and the profound significance of nuclein, now known as nucleic acid, became evident. It wasn't just the building block of genes, but the carrier of hereditary traits. Miescher's nuclein exists in two forms: DNA, or deoxyribonucleic acid, and RNA, or ribonucleic acid. In most organisms, including us humans, these nucleic acids combine with protein to form nucleoproteins. Here's the key insight: hereditary information is encoded within these nucleoproteins. Remarkably, this information uses the same substances to replicate itself and produce more nucleoproteins. DNA chemical and physical properties make it perfect for both replication and information transfer. Each DNA molecule consists of two long intertwined strands forming a chain of subunits called nucleotides. Every nucleotide includes a sugar, deoxyribose, a phosphate group, and one of four nitrogenous bases: adenine, A, guanine, G, thymine, T, or cytosine, C. The sequence of these nucleotides encodes the genetic instructions, dictating the order of amino acids in the polypeptide chains that form proteins. These A, G, T, and C nucleotides are sometimes called genetic markers. Together, they make up the genetic code or human blueprint, essentially mapping human nature. Like much in genetics, these markers exist in diploid base pairs, one set from dad, the other from mum. Their specific sequencing determines nearly everything about our genetic inheritance. Humans have approximately 3 billion of these base pairs, collectively known as the genome. The effort to fully map this genome is called the Human Genome Project, or HGP. These base pairs enable genetic identification and make genetic engineering feasible. By blocking, enhancing, or replacing specific base pairs, a process called genetic therapy, scientists can alter genetic outcomes with growing precision. In the United States, Congress approved and funded the Human Genome Project in 1988, launching an ambitious 20-year endeavor. But challenges quickly emerged: internal rivalries among geneticists, debates over costs, and resistance from various vested interests. Despite yielding more data for scientists, philosophers, and sociologists than the moon landing, the project faltered in the early 1990s. It was hastily revived when news broke that Japan had started its own genome project 5 years earlier. Even with this renewed effort, the US lost valuable time. Having begun late, it stands little chance of outpacing competitors in the race to complete the genome map, the fundamental blueprint of humanity and a gateway to possibilities yet unknown. Thanks for joining me on this genetic journey.